Okay, welcome to the course of Comprehensive Molecular Diagnostics and Advanced Gene Expression Analysis. So, this is the week 3 discussion that we are having, and this course, this week's videos were more on DNA cloning. So, we had DNA cloning and recombinant DNA technology, and also and on the later parts of the videos we got to see a lot of uh, genome editing applications. Yeah, so okay, so we have some people joining in. So okay, we actually I also we discussed one question here last time, so maybe you people also can when in the end maybe we can discuss that too, if you have done. So, about this RDT or recombinant DNA technology, we saw some of the key components here. Yeah, anyone want to speak anything? So we saw some key components in which one was restriction under nucleases, which were which came out to be molecular scissors that can cut DNA at specific sites. Then we had vectors in the form of plasmids, bacteriophages, cosmids, artificial chromosomes. Yeah, and then we had some host organisms, bacteria, yeast, and several of mammalian cells too. The process is the common process that is involved is basically of cutting the DNA with restriction enzymes, inserting the DNA fragment into the vector, and then introducing recombinant DNA, that is the new DNA, into a host cell, and then select and grow cells containing the recombinant DNA. So, in that, we also learned about DNA libraries, in which we had several genomic libraries, in which it usually means represents total chromosomal DNA. It contains coding and non-coding regions, and they are prepared by digesting genomic DNA and inserting fragments into vectors. Yeah, and then you have cDNA library, in which it represents mRNA from a specific cell or tissue. Those contains only exons and are prepared by reverse transcribing mRNA to cDNA before cloning. We also have some screening methods: colony hybridization, PCR based screening, and hybrid selection. We learned about some genomic editing tools also, the zinc finger nucleases, yeah, the talents, and the CRISPR-Cas9. So CRISPR itself is a big wing, in which how it want we can the CRISPR-Cas9 we can talk in detail, in which there are several key components. Yeah, the Cas9 nucleus itself, and the guide RNA, which involves the crRNA plus the track RNA, and the PAM sequence. So the mechanism it works is that the guide RNA directs Cas9 to target sequence, and the Cas9 binds and cleaves DNA near the PAM site. DNA is repaired by NHEJ or HDR, introducing edits. So it has now applications in gene knockout, disease modeling, therapeutic gene editing, and also in diagnostics. So nowadays, actually, this CRISPR-Cas emerged as a genome editing tool, but it has now have seen a lot of interest in diagnostics field as well. And it has been very too much appreciated for detection down the level of single mutation detection too. Single nucleotide polymorphism is also something that CRISPR can pick up. Okay, we'll go to the problem discussions now. Which phenomenon is exploited in blue-white screening during molecular cloning? So we'll wait for maybe 4 or 5 minutes. People can answer your question answers in the chat box, and then we'll discuss. So the answer is LAGZ gene expression. So blue-white screening in molecular cloning explores the expression of LAGZ gene, which encodes the enzyme beta galactosidase. Yeah. So this technique is used to identify successful recombinant clones during the cloning process. Yeah. So you people know, yeah, how blue-white screening works. Yeah. So this cloning vectors, it contains multiple cloning sites, MCS, with the LAGZ gene. So when a DNA fragment is successfully inserted into this MCS, it disrupts the LAGZ gene and preventing the production of functional beta galactosidase. Yeah. So the bacterial clones colonies are grown on media containing X-gal, a colorless. It is a colorless substrate. Yeah, which turns blue when cleaved by beta galactosidase. So whichever colonies which have successful insertions will appear. So basically, colonies which are recombinant will appear white because they cannot produce functional galactosidase to cleave this X-gal. Yeah. So and colonies without insertions will appear blue because they can produce functional beta galactosidase. So this is a way of screening, as we discussed. Is it clear for all? Okay. what distinguishes a genomic library from a cDNA library in molecular cloning? Genomic libraries contain only introns, while cDNA libraries contain only exons. Genomic libraries are made from mRNA, while cDNA libraries are made from genomic DNA. Genomic libraries represent the entire genome, including introns and exons, while cDNA libraries represent only expressed genes. Genomic libraries are constructed using PCR, while cDNA libraries are cDNA libraries use restriction enzymes. So the correct answer is C. Yeah. Genomic libraries represent the entire genome, including introns and exons, while cDNA libraries represent only expressed genes. So genomic libraries are constructed using the total chromosomal DNA of an organisms, which include coding regions, basically the exons, and the non-coding regions. Just wait. Some people are entering now. So genomic libraries are constructed using the total chromosomal DNA of an organism, which include coding regions and non-coding regions, basically exons and introns. Also, in non-coding regions, you can include introns, promoters, and regulatory sequences. And then cDNA libraries, on the other hand, are made from mRNA through reverse transcription. Yeah. So they represent only the expressed genes, exons, of a specific tissue or cell type at a particular time, excluding non-coding regions. Yeah. So introns are excluded in cDNA libraries. So C is the correct. Yeah. Is it clear for everyone? Perfect. Which method entails the selective hybridization of mRNA with specific probes, enabling the isolation of mRNA molecules carrying the target sequence for subsequent translation in vitro in screening in gene libraries. So the answer is hybrid selection translation. So this method uses DNA probes from recombinant clones to hybridize with complementary mRNA, which is then isolated and translated in vitro to identify clones containing the target sequence. Yeah. So in this, what this technique is essentially used to identify and isolate specific mRNA molecules from a gene library. So in which you can actually identify clones carrying the target sequences by, as it says, selectively hybridizing mRNA with specific DNA probes. So what it usually has some steps here. First, so if you go for this hybrid selection, first you will prepare the prepare the probes. Yeah. So as it written, DNA probes on recombinant clones. Yeah. Then what you can do is you have to hybridize. with mRNA. Then you have to go for washing and elution. So unbound mRNA will be washed off. So only the hybridized mRNA bound to the DNA probes will be left, and you can then go for in vitro translation. And after this in vitro translation is done, so you can go for protein analysis. In which the translated proteins, you can analyze those using techniques like electrophoresis or immunoprecipitation, so to detect the protein of interest which correspond to the target mRNA. Yeah. So the hybrid selection translation is the correct answer. Okay. Dash involves the transfer of DNA from bacterial colonies onto a solid support, typically a membrane. Following transfer, the DNA on the membrane is denatured and then hybridized with a labeled probe specific to the target sequence of interest. Through autoradiography or chemiluminescence, the presence of the desired DNA sequence can be visualized. Okay, I think the answer is southern plotting or what? Plant colony hybridization. Okay. Yeah, the answer is colony hybridization. So it's a technique in which you will identify bacterial colonies and containing specific DNA sequences or genes of interest. Yeah. So in this, so you have to prepare the master plate. So in which you have bacterial colonies in which will be cloned. So the steps will be the master plate preparation. Yeah. So you will glow the colonies and then you will put a replica plating on nitrocellulose membrane. Yeah. So the nitrocellulose filter is basically pressed onto the master plate to transfer the bacterial colonies onto the membrane. So it will create a replica of the original plate. Then you will go for cell lysis and DNA denaturation. So cell lysis and DNA denaturation. Yeah. So this can be done uh, by like treating the nitrocellulose filter with detergent like SDS, such that it will lyse the bacterial cells, and alkali also to denature the DNA into single strands. So making it available for hybridization. So then what you can do is, as it is written, you can have fixation, yeah, using UV. So usually temperature, you can also go for temperature, or UV is also UV cross linking also there, and then you can go for uh, labeled hybridization. So hybridization with labeled probe. Yeah. So a labeled probe, which can be as it is written, auto radiography, then it will be a radioactive label, or it is fluorescent or chemiluminescent, anything that is complementary with the target sequences added, and then the probe can directly bound. Next step, it's usually washing, yeah, washing, and then the last is detection or identification, whatever I can say. Okay. Cool. Which statement accurately describes the characteristics of zinc finger nucleases? So the answer is C. Each, each zinc finger recognizes approximately 6 to 8 base pairs within DNA. So zinc finger nucleases are engineered proteins composed of two domains: DNA binding zinc finger domain, which recognizes 3 base pairs, and foci, a nucleus domain for DNA cleavage. Yeah. So the zinc finger nucleases are These are engineered site-specific endonucleases used for genome editing. Yeah. So as I told, the two domains, yeah, DNA binding domain, and and then you have the fork1 nucleus domain. Yeah. So they have a mechanism in which these can bind to a specific DNA sequence based on their amino acid composition, which determines their affinity for particular base pairs. And then you have some form of dimerization, and so two ZFN proteins bind to adjacent target DNA sequences, bringing their foci nucleus domains close enough to dimerize. This dimerization activates the nucleus, which cuts both DNA strands at the target site, creating a double-stranded break. And then you have the repair mechanisms like the non-homologous end joining or the homology-directed repair. That's what how the gene editing works here. Which genome editing mechanism relies on the cell's repair machinery to incorporate exogenous DNA sequences into the genome? So the answer is HDR, or homology directed repair. So HDR is a DNA repair mechanism that uses a homologous template, including exogenous DNA, to precisely incorporate specific sequence into the genome editing, into the genome during genome editing. Yeah. So the other options are essentially the tools like CRISPR-Cas, ZFN, or TALENs that induce double-stranded breaks at specific sites, which are then maybe repaired by HDR for targeted gene editing. Yeah. So C is the correct answer. The CRISPR-Cas9 system originated from. So the correct answer is bacterial immune system. Yeah. So CRISPR-Cas9 is derived from bacterial adaptive immune systems that protect against invading phages or plasmids by recognizing and cleaving foreign DNA. This system uses short palindromic repeats and Cas proteins to store memory of previous invaders and target them during subsequent infections. Yeah. Okay. Kind of a repeat. In a restriction digestion experiment, you obtain the following fragment sizes after separating them on agarose gel. So these are the sizes. Calculate the total size of the original DNA molecule. If this was the partial digestion of a circular plasmid, what would be the minimum size of the plasmid? Answers? So, to get the original size of the DNA molecule, what we can do is we can sum up the fragments, all the fragments. Yeah. So 23.1 plus 9.4 plus 6.6 plus 2.3 plus 2 plus 0.56 plus 0.12. So the answer is 48.48. Yeah, kilobits. And answer of B, if this was the partial digestion of a circular plasmid, what would be the minimum size of the plasmid? So remember, it is asking for the minimum size here. So, so as it is written here, if this was a partial digestion of a circular plasmid, the minimum size of the plasmid should be equal to the total size of all fragments. Yeah. So the minimum size also will be 48.448 kilobase per. Yeah. So to note that this is the minimum size. The actual plasmid could be larger. Yeah. Correct. Okay. Yeah. Okay. A plasmid was digested with enzyme HPA2 on agarose gel a plasmid was digested with enzyme uh, HPA2 on agarose gel electrophoresis. You observe three bands: 100 to 230 and 500 base pair. How many HPA sites are present in this plasmid? What are the distance between each site? And what is the size of the plasmid? So, okay, a lot of people have solved this. So, we also have a second part of it. Okay, a second cut of the plasmid with BAM H1 yields two pieces, 80 and X base pair. How many BAM H1 sites are in the plasmid? What is X in base pairs? Can anyone explain the first part? Maybe someone. Can any of you explain? Okay. Try to draw it. So you are getting three bands, yeah. So basically 100 to 30 and 500. Yeah. So what happens is what? So maybe we can do it like yeah. So here you how many HPA two sites are present in this plasmid? So it will be as you are getting three, it will be three. Yeah. So basically, if I have if I have one cut, I will have one, yeah, I will have one product, yeah, because it will elongate, it will become straighter, and if I have two cuts, I essentially will have two pieces, and definite, and similarly, if I have three cuts, then I will have three products, yeah. So yeah, three we have. So okay, so maybe I can put this one as 100 base pair, yeah. Then you have 230 base pair and 500 base pair. So what are the distance between each side? Yeah, that you can calculate using the difference in the base pairs. Yeah. And what is the size? Size we can calculate. So yeah, distance you can calculate. This is 500 base pair. This is 100 base pair, and this is 230 base pair. Yeah. And total the size of the plasmid is 100 plus 500 plus 230 base pair, which is 3 830 base pair. Okay. Yeah. Okay. Yeah. So that's how better is to draw and solve it. That's better. So coming to the next part. So a second cut of the plasmid with BAM H1 yields two pieces. Okay. So how many BAM H1 sites are there in the plasmid? So this BAM H1, it is cutting. It is putting two pieces. Yeah. So definitely you have a X base pair, yeah, and then you have a 80 base pair. So you have here it will be 2, and what is the X in base pairs? So it will be So total we know is total, we got that total is 830. Yeah. So what is the x? X will be, so 80 plus x will be 830. Yeah. So x will be 830 minus 80 base pair. So 830 minus 80 is 0, 5, so 750 base pair. Okay. So that's the B answer. Yeah, B answer B is yeah, that's the answer. Yeah, you will get in terms of base pairs only you can tell what are the distance between each side. The 100 and 230. So these are the cut sides. Yeah. Yeah. That's the distance between them. Yeah. So anyone did this question? Okay, others? Okay, one of you have put the answer. Okay. Okay, I'll give 15 minutes for people who have not tried, and then we can discuss. So what is the formula for Tm? So Tm you can calculate by Tm equal to 4 degrees Celsius into Gc plus 2 degrees Celsius into 80. So yeah, I'll tell. So yeah, so as I told, as we discussed, there can be multiple answers for this. Yeah. So I had for forward, one example is 5 prime. I have G G A G C T T T G T T T C A A C C A A G. This is the forward primer, and for reverse, you have A T T A A A T G C G G A A T T G C C C A. Okay, so these are two examples that I am giving you. So the product I am getting from it is 131 base pair long, Now you can calculate. Can someone calculate the TM for this? I have not calculated TM for forward and okay. How many A's we have in the front? Okay, one, two, three, four, five. Okay, five A. One, two, three, four, five, six. 6T. One, two, three, four, five. 5G. One, two, three, four, 4C. Yeah. Okay. So 4 into G is 5 and C is 4 plus uh, 2 into how many we have? A is 5 plus T. T is 6. So 4, it's 9 plus 2 to 11. Yeah. So we got 58 degree Celsius here. Similarly here have how many is it counted here wrongly? How many A's are there? 4, yeah, 4 it is. 1, 2, 3, 4, 5. Yeah, 5. Okay, okay. Sure. In the reverse, 1, 2, 3, 4, 5, 6, 7. 7A. 1, 2, 3, 4, 5. 5T. Then you have 1, 2, 3, 4. 4G and 1, 2, 3, 4. 4C. Yeah, correct. So similarly 4 into G is 4 plus 4, so G, C, and then you have 2 into A is 7 plus 5, so A, T. So you have 4 plus 4, 8, 4 into 8 plus 2 into 7 plus 5 is 12. So 32 plus 24. So 4 plus 56. So 56 and 58. So this one is 58 degrees Celsius and this one is 56 degrees Celsius. Okay. Then you can also calculate G plus C percentage and stuff like that also. Yeah. So it's only the total number by basically all the nucleotides divided by only G plus C into 100. So like here, if you want to calculate G plus C percentage, how to do it? Can you calculate for the first one? So you have here how many G? So you have 5. So this total is 5 plus 6, 11. 11 plus 5 is 16. Yeah. So and 16 plus 4 is 20. Yeah. So 20. So 20 divided by 5 plus 4, 9 into 100. Yeah. So 9 divided by 20 into 100 is 45. Yeah. So 45%. Yeah. And similarly here it will be 7 plus 5 is 12. 12 plus 4 is 16. 16 plus 4 is 20. So for 8 by 20 into 100. So this will be 40%. Yeah. Okay. Then yeah, that's all.